Great. So first, thank you for the invitation, and it's a great opportunity to be here, share my recent work. And my name is Changhan. I'm the third year postdoc, and since I'm teaching this quarter, so they switched my title to the business assistant professor. But yes, basically it's the postdoc and in lab. And the title of my talk today is identifying external communication signals and analyzing their associated signaling networks using expounder. Before moving forward, step down so you can hear it. Okay. Yeah. Okay, great. So first, we basically interested in cell development. So this is a very general, very big topic. And so the critical processes for cell development is including cell proliferation, migration, differentiation, and programmed cell death. So these critical processes, that's one of the major reasons that the cells can have these critical processes is the ability of a cell to send, to receive, process, and transmit the signals with its environment and with itself. And then this very straightforward question that how the cells that are sending signals to each other or receiving signals from other cells and the environment. And the basic mechanism of cell-cell communication that we start from. So for a cell, we have a receptor, and then once we found the corresponding ligand, the ligand binds the receptor, and then the transduction turns on, and then the signal transduction starts from the receptor through the transcription factor. So we call a TF here through TF, and then the response will be transcribing the DNA. So these three steps can generally describe the process of cell-cell communication. And so these three steps of cell-cell communication include the first, reception, the ligand receptor binding, and the second step, that's the transduction, receptor to TF interaction. And then the third part, that's the response, the TF regulated target gene expression. And so then what's the external signals? So basically, usually we are looking into the measured cells that we take some cells in the lab and then do the measuring, do the sequencing, and then we get the transcriptomics. And we have a lot of different methods, like computational methods, can analyze this data and then can infer the cell-cell communication within these measured cells. And what if we have signals, ligands that are coming from unmeasured cells, or coming from like induced signals that manually add by in experiments, and or the extracellular signals. So these signals that we call that external signals. So the communication between the measured cells and the external system, which may include the unmeasured cells, extracellular signals, or induced signals, plays important roles in functions of these measured cells. For example, there's a paper that during the experiment they manually added these three ligands, TGF-beta1, EGF, and the TNF, and these three are not expressed in the measured cell, and then they all can serve as promoters during the EMT this transition. So this is a very good example showing that the external signals can play a very important role in the functions of the measured cells. However, the current methods only deal with the ligand regulated signals sent by the measured cells in the data. So that is the signal must be related to a ligand that's highly expressed in the measured cells. So on the other hand, if a ligand that is lowly expressed, that not very high expressed, or even not measured in the measured cell. So the current methods cannot identify this ligand. So therefore, the motivation is that how can we just develop a new method that can using what we have, using the data from measured cells to infer potential external signals. So this is the method that we call exFINDER. So the paper is published earlier this year on NAR. And then we firstly here, I will give a brief introduction of the workflow for the exFINDER. So first part is that we have the inputs of this method. So first, the two required input, one is the expression matrix. That's the data from measured cell. And then we require the cell cluster labels. And then there's another information we need to manually input, that is user select the target genes. And compared to current methods, usually we start from the ligand and then explore the downstream genes that's from the top to the bottom. And then here, basically, we select the target genes first. That means we start from the bottom and then we goes up. So here, and then there are some optional options that we can also input. So first we can discover the marker genes, and then these marker genes may help us to select the user interested in genes. For example, if we are focusing on like what signaling pathways can activate these clusters, then we can select the marker genes of these clusters to be our target genes. And then trajectory analysis can also provide helpful information that can help us to look for the possible signals and possible sender cells. And then also the last one, critical transition analysis. So this one is corresponding to this paper published last year, also on NAR. So this paper that introduced a method, and there's a package called biotip, TIP, biotip. So this method also can be used to perform critical transition analysis. So critical transition, for example, cell differentiation can be regarded as one of the critical transitions. So this method can help us to identify the group, that the cell population group that has the highest index of criticality. So that's means that cell population group is highly undergo the critical transition. So these are the part B is just some optional information. And then once we have part A prepared, these three inputs, we can first perform the first step of exFINDER analysis. So first we infer the ligand target gene regulatory network using literature or experiment supported database. So this database, we did not manually collect the data. We integrating several publicly available databases, including the databases of NicheNet, CellChat, and other two. So basically the basic framework is based on NicheNet database, because NicheNet database already has these three layers. And then our exFINDER database is kind of like improved in NicheNet database, and then also include the three layers. That's from ligand to receptor, from receptor to TF, and from TF to targets. And then also the rest of the database are mainly about human, and then exFINDER database is available for human, mouse, and zebrafish. So the database mainly include three big tables. So one table for each layer, and then start from the target. We can use the TF target database. That's the last one. We can use this one to infer the corresponding TF that's interacting with these target genes. And then we can use the second table to infer the corresponding receptor, and then the last step that's from the receptor to infer the corresponding ligands. So that's the basic logic that we do the inference from the bottom to the top. And once we have this ligand to target GRN, we can identify the external signals using the single cell data. We can, for example, the target genes is highly expressed, and then their TFs and their inferred receptors are also highly expressed, and so there has to be some signals they received somewhere. If we did not find the ligand expressed in the measured cells, it might be the ligand come from the unmeasured cells or the external environments. And then based on this heat map, that's the we identify the external signals. Then Expounder can convert this network. We call that external signal related signaling network. So that's exSigNet. So this network, we convert this network into a directed graph. So this graph, which has nodes, so each node is a gene, and then we have edges. So each edge represents interaction between two genes. So that's the first half of the Expounder analysis. So the second half of the analysis, including different parts. So first the parts, we have the nodes, we have the edges, so we have the directed graph, and then for each node we have the gene expression. So basically we can predict the signaling strength for each interaction. So each interaction has two genes, so each gene has its own expression. So we use the mass action law. So that's very basic, but it's reasonable. So we use that to predict the signaling strength for each interaction. And then after that, so in the left panel, we saw these graphs. This graph can show the interactions, and then the size of the node can show the expression level for this gene, and the color for the edge shows the predicted signaling strength for this interaction. And the next part, we can predict the dominant signals and the targets in this network. So how we do that, we're predicting the dominant signals and targets based on the maximal signal flow. For example, here is a signaling network. We select one target, and then we select one ligand and one target, and then we will have a path, or maybe a lot of paths to connect these two. Here it's a very simple network, so we saw that this just one receptor and one TF, so this is just a chain. But usually we have multiple receptors, and a lot of TFs can connect these ligand target pair. And then there's a basic, we can convert that into a math problem that calculating the max possible flow between these two. So then we, based on this maximum signal flow, we can calculate which ligand has the highest signal outflow and which target has the highest signal inflow. So that's how we predict the dominant signals and target. And then for each signal target pair, we can have a pathway. And then for a big network, we can have a lot of ligands, a lot of targets, and then we will have a lot of ligand target pathways. And then for these pathways, we can do clustering, and then we can show which ligand target networks have the similar structure, and then we can look into the clusters and see if they have a similar biological functions. So these are the second part of the Expounder analysis. And then after that, the third part, we introduce a new concept that's an activation index. So activation index is that for one exSIGNet, so for one big network, we use this formula to quantify the activation level for this network. Basically, the first term includes the total number of nodes and edges for this network. So first term consider the general size for this network, and the next three terms describe the total signaling strength for 3 layers. That's like to receptor to TF to the target is 3 layers. And then we combine all these factors together. We quantify the activation index. So this activation index like enable exFINDER to compare 2 different exSIGNet, so 2 different networks, and then to compare these activation index, activation level. And then also in the middle panel, so this is how we connect expounder to the biotip method that can the output. One of the most important outputs from biotip is that it can return to us the important TFs that corresponding to the critical transition, and then expounder can take this information, so infer the signaling networks based on these like critical transition signals. That's these TFs, and then we can see that how these important TFs work and what are the target genes that these TF activates. And the last part is that we can evaluate the goal analysis result. What we do is that we input the whole inferred external signal target network, or input this whole network, and then do the geo analysis, and then we can have different terms. So we use we compare these terms. For each term, we can have a complete gene list for each term, and then we compare these genes to our network, and then keep evaluate these the top 5 geo analysis results. So that's the these three parts are the main analysis parts for expounder. And
Can I ask a question?
Yeah, sure.
So for these analyses in particular, you're talking about like doing it on a specific network. So is the network here like, I just need an idea of what the resolution of the network is. Like, is it on a specific, like everything that's going on in one cell type, or is it like a specific pathway that's happening? Like, where are you drawing the line or the distinction between discrete networks in this case?
That's a great point. I think currently, so the network is based on the resolution is based on cluster to cluster. So that's why the input we need cell cluster labels. So we so for example, we have this very simple network as an example. The target is maybe like cluster 3, and then the ligand come from somewhere else, and the receptor and TF come from cluster 2. So that's basically yeah, we are talking about the communication between clusters.
Okay.
Yeah. But that's a very good point. We are still working on developing a new method, try to have a higher resolution, even for single cell resolution or a customizable resolution.
Okay.
But yeah.
That was gonna be my next question. Could you like go even further and say, okay, what's the your activation index for like one pathway in the network, or what's the activation, or sorry, what are the go terms associated with one pathway in the network?
Yeah. I think right now we does not include that part in expounder.
Okay.
Yeah, but yes, I think
right. I mean, obviously that's an overstatement, but like it's not just a one-to-one relationship here.
Yes. I think I talked about your point before with other people. I think is one of the challenging thing is that for pathways we need to like build a bridge between the infer network to the current public data for these pathways. For each pathway, we may have a lot of genes, and then some are TF and some are just maybe small molecules. We don't have the much information about that. And then once we do that, we can move forward. We can combine the single cell RNA data, but we still need maybe protein data, proteomics data to go one step further to confirm these genes actually already like go to the protein level and then really be activated. But yeah, I think that's also part of my ongoing research. Yep. And then the first part, that's so in the paper, that's for benchmarking, we use the Expounder to compare with other two methods. One is for cell chat, one is iCellNet. So these are, so in the middle panel, this graph basically means that, so how many, like what's the percentage of the ligand receptor pairs that inferred by these two methods can be also recovered by Expounder. So the on the right hand side, this column, lastly, we only use the Expounder database. We do not use the single cell data. We only want to test the comprehensiveness of this database, and then we found that most of the inferred ligand result pairs can also be recovered by Expounder. And then for this for the main part, these are some cell population group specific communications. That means, for example, these LC specific communication, that means the ligand only highly expressed in the LC cells talking to this APOE plus FIB cell. So that means the ligand only expressed in the LC specific cells. So this means Expounder can fully recover these cell population group specific ligands, and also on the other hand, if we remove this cell population cell group from the data, the rest of two methods will not be able to identify this ligand, because the rest of two methods only can like do the cell-cell communication analysis based on the measured cells. And then the third panel, that's the example for Expounder and inferred external signal. So for if we are looking for the signal network that activate the target genes, the marker genes of the FBN1 plus cell group, this cell group. So the target genes are the marker genes for this cell group. And then we found out that there's one ligand, that's APOE, only expressed in APOE plus cell, and then it still can activate these target genes. And so this pathway related to external signal, for example, if we remove this cell group, APOE cell group, and then the current method will not be able to identify this ligand. So these are the benchmarking part. And then the next part, we apply Expounder to the zebrafish kernel neural cluster cells. So these zebrafish cranial neural cluster cells are have a very high heterogeneity during migration. So this data comes from our collaborator, Dr. Schering, his group. So this is I think this paper published on 2019. And then what they did is that they measured neural cluster cells at different time points. For example, here we the middle panel, you show there are 6 different time points, and then these are the single cell RNA data. and then they label the cell into 5 different groups. So the interesting thing is that in their experiments, they show that besides neural cluster cells, they still have 1, 2, 3, 4, yeah, at least 4 cell groups that are non-neural cluster cells. And then in the original paper, they only analyzed the neural cluster cells. And first we did the cell chain analysis for all these cell groups, including the neural cluster cells and non-neural cluster cells. We saw that there are very strong communication between these cells. So it's possible that these neural cluster cells can receive some signals from these non-neural cluster cells. So if that's true, that means in the later analysis that we should include in these non-neural cluster cells. And then here, first part is that expounder identified one ligand. So this ligand only highly expressed in epithelial cells. So that's this brownish group. So this ligand highly expressed in this group and then has a very low expression in the neural cluster cells. And then if you perform regular cell-cell communication analysis to these neural cluster cells, you will not be able to find this ligand. And then so expounder actually found this ligand. We just plot this heat map to prove that okay, this ligand actually did highly expressed in this cell group. And then expounder shows this signaling network. That's this ligand talk to the receptor and then to the purple nodes dots represent the TF, and then the blue dots represent the target. And then we also show the predict signaling strengths using this color scale. And then we come back to the single cell data. We also confirm that yes, this gene is highly expressed in and only in the epithelial cell. So this ligand receptor interaction is also supported by recent studies, and you can find the information in the Expounder paper. So this is the first part, and we do this part because we like we use these non-neural cluster cells, these cells. We have the data. We can use that to support our expounder analysis result. So we can find okay, this highly expressed and this highly expressed in the cells in this group. And the next part is that we actually found another ligand. So this ligand has a very low expressor for all cell groups, like both neural cluster cell and non-neural cluster cell. So we only can say that this inferred ligand receptor interaction is consistent with a previous study, but experimentally we do not have data to prove that. But based on the expounder analysis, we think this one should, like, the cells should receive these signals somewhere else, because the receptor is highly expressed and the TFs are also highly expressed and the target genes are also highly expressed. So yeah, that's the basic logic. And so see that this ligand is not highly expressed in all measured cells. So this part is that we performed the biotip analysis. We found that these two TFs are the very important TFs related to the neural cluster cell differentiation. And then Expounder can show you the corresponding signaling network related to these two TFs. So we found that, okay, these TFs actually activates both the target genes for both two cell groups. So they activate these two marker genes for the pigment cells. these three marker genes for the skeletal cells. And if you come back here, the pigment cells and the skeletal cells are the differentiated cell groups. So it's coming from the early neural cortex cell and the transition cell. So expounder can show you the corresponding signaling network related to these important TFs, and then support the finding. So that's I think that's the first application for expounder. And also we apply expounder to the mouse data set. So in this data set, we have a more complicated case. For example, here, in the original data, the author pointed out that so these two groups will merge at some point. So these are very noisy cells. So and then we have two branches, branch A and branch B. And basically, so in our expounder paper, we summarize these profile. So we can say that basically we have two branches. So first, that's the late NC cells, and then boundary cap cells, these two cell groups. And then we have, in the paper, they call this unassigned cell group 1. So that means these cells are very noisy, possible transition cells. And then we have two different cell groups, and then later this goes to two directions. So first we perform a biotype analysis. We show that, okay, these late NC cells are undergo a critical transition. And then we select the marker genes of this cell group as our target genes. And then, so the marker genes are the target genes are these blue dots, and the expounder found that there are three possible external signals that can activate these marker genes. So let's activate the marker genes of the late NC cells. And then interestingly, all these three ligands are corresponding to three interactions, ligand-receptor interactions. And all three ligand-receptor interactions are supported by existing studies. And then expounder showed us that which one has the highest total signal outflow. So PLD2, this ligand, so this signal has the highest total signal outflow. So compared to the other two, so this is the dominant external signal. And then compared to all so comparing all these target genes, and we saw that the GIS1 has the highest total signal inflow. So that means these may have the highest activation rate, or like we can say these received the most signals from within these signaling network. And so also on this data set, that expounder can predict uncovers the transition path. For example, as shown in this picture, so these are the dark blue cell group. So it can come from this unassigned cell group 1, or this can come from this like the light blue, the boundary cap cells. That means since these two branches kind of merge in the middle, so it's not very clear that this group come from the unassigned cells or come from these boundary cap cells. And so these are the two possible paths. Then also in the original paper, the authors point out some early divergence genes along branch B. So that's this branch B, and their related TFs. Then we perform expounder based on these early divergence genes. We set these early divergent genes to our target genes, and then their related TFs to the corresponding TFs. And then we use this information to infer the possible signals and the receptors. And then we found the interesting thing is that we have a lot of ligands that talk to these two receptors, and then these two receptors later activate these TFs and the target genes that provided in the original paper. So that's then since these ligands activate these two receptors, we check the expression level for these two receptors in these two cell groups. So we found that it's more likely, the more signal coming from this unassigned group, but it's also possible that there are signal coming from the boundary cap cell. So this light blue group. So that means these two paths are all possible. And another interesting thing is that these IF and NR1, so all these all these on IFNA, so this receptor IFNAR1 is the receptor of 12 subtype IFNA genes. So these IFNA genes. So these are so this is the information from previous study, and also the interferons are acted directly on these cells to produce pan sensitization. So this is also support our expounder results, and then we can input this whole network compared to the goal term analysis result, and then provide us the top 5 goal terms. So these are the second application that we present in the paper. So also the last application that's also very interesting. So this is about the manually added inducers. So this paper that I mentioned before, they in this paper, a comparative analysis was performed for the EMT response with 3 different added inducers, and the single cell data was used to measure the expression profile of 4 human data sets. So this is very interesting because, for example, if we use one of the data set, A549 data set, and then we perform exFINDER analysis, so hopefully the expounder should capture one of these manually added inducers. So these added inducers are perfect examples for external signals. And then, for example, for the A549 dataset, so target genes is the EMT regulators provided by the original paper. It's a typo here. So we use these EMT regulators as our target genes, and then for we present 2 cases here. We have more on the paper. So for 2 cases on the left-hand side, this case is that TGF-beta1, that's the manually added inducer, and the right-hand side, that's TNF, is the added inducer. So on both cases, expounder can find this external signal, although they have a very low expression in the data set, and even for TNF, it has no expression. It's not even measured. It's not even measured in the data set. So expounder can identify these 2 external signals and then infer their corresponding signaling networks. For example, for this one, we show that ITGB1, this is the receptor, and then if we check the expression level, we saw that also after adding the inducer, the expression level for this receptor goes up. Now for the rest of the 2, they are relatively stable. It's not very easy to tell. The difference. But at least the expounder were able to identify this external signal and infer the corresponding signaling network. So these are the main part for the paper. The summary is that exFINDER may be the first computational method to identify external signals in cell communication and infer the associated signaling networks and do some quantitative analysis. And then the central to this method is the exSigNet, that's the external signal related signaling network, which contains the path and the nodes that represent the genes and the edge weight represent the predicted signaling strength. And the corresponding R packages are available online. You're all welcome to test that. I'm still updating the database. Yeah, I think that's the main part for the paper. Do I have time? Cool. Yeah, I think next part is that I can talk a little bit more about our ongoing research by combining Expounder and several other methods. So basically, the basic idea is that this is a recent review paper based on neural cluster cell development in zebrafish. But the general idea is that usually we talk about differentiating. We are referred to very important mathematical concept that's a bifurcation. So that means from one cell type to two different cell types. But is that always the truth, or is this the best explanation for cell differentiation? Sometimes we may see that that's from one cell population cell group to more than two cell groups, maybe 3 or 4. So they give a new name like a multiplication. That's from 1 to 3 or 4. And then also we have different types of possible bifurcations. So these are the concepts that presented in the review paper. And then we want to know that, recall that in the Expounder paper, we use the zebrafish neural cluster cell data. And we go back here. So in this data, all these cells that are identified as transition cells because these cells are very noisy. But can we just put that, or is this the best way to put that all together, name that to one single group? So we want to know if they should be identified to different subgroups. So if we can identify that to different subgroups, so we can discover multiple transition states. So this is the research on this area is still on the early stage, but will lead us to very interesting results. So these are the experiment results showing that the transition groups are very noisy, has a little bit of everything. And then there's another paper that published by Paige. He's a former postdoc in Near Lab, and he's now assistant professor in Peking University. So the name of this package is called MuTrans. So MuTrans is like can distinguish stable and transition cells. So this method already be used in another experiments that published in the result has published in Cell or in Nature. And then the general idea is that he used a new new concept called entropy. He used the entropy to quantify to distinguish the stable and the transition cells. So the cells with higher entropy will more likely be transition cells. And the cells with very low entropy means they are very stable. And for example, here they're very stable and they're more likely to become stable cells. And then, so he has all these in this paper published in 2021 in Nature Communications. And then we performed this his method, and we found out actually at least we can identify 2 transition groups, and then we have multiple early neural cluster cell subgroups and multiple skeletal subgroups. And to confirm that, we actually found several marker genes to mark these two transition groups. And then for these 5 skeletal subgroups, we also found very interesting marker genes to separate these subgroups. And then some of the marker genes are really related to, later we found out that some of these marker genes are related to another very important phenomenon in zebrafish group. That's the dorsal ventral patterning. So some of these genes are just region-specific genes. And then the results shows that the general landscape for the transition from the early neural process cell to transition cell, and then we may have multiple transition states, and to pigment, skeletal, these represent subgroups. So you see the cells on the on the lower level. That means these cells are very stable. So the transition cells are in the middle. So it's a relatively high entropy level. So it's not very stable. And then, so we check if some of the genes are dorsal ventral related to dorsal ventral patterning. So these are the patterning during the zebrafish growth. And then, so I will skip this part. This is another paper shows some results related to the dorsal ventral patterning genes. So actually, we found some genes are the dorsal ventral patterning genes. Some genes are region-specific genes, and then we perform Expounder, and for example, these genes are highly expressed in these skeletal subgroups, and then we want to find if there's some signals can activate these genes coming from like the neural cortex cells or non-neural cortex cells. And then we found that there are at least 1, 2, 3, 4 ligands that from non-neural cortex cells, and then 1 ligand coming from the neural cortex cells and the transition cell. So these also can confirm that the can support that we have multiple transition states, because these ligand only highly expressed in one transition state, and then in the other transition state it has very low expression level. So I think that's the main part of my talk, and the ongoing research that I'm doing right now is to first improve and refine the database, and then try to have a new method to infer and analyze the signal pathways at a higher resolution, maybe single cell resolution or a more customizable resolution. And then we try to have the new methods can utilize the spatial and time information. That means if you have spatial information, you can assume that these two cells are too far away, right? Even if their ligand receptor are highly expressed, but it's impossible for them to talk to each other. And then time information, because if you have 2 time points, one is 12 hours, one is 24 hours, these 2 time points are so far away, they have 12 hours gap, so they cannot talk to each other. So we try to utilize this information to refine our inference results. And then last part, thank you for the funding and for my advisor, my collaborators, and yeah, that's all for my talk. Thank you.